Okay, good morning, everybody. I think we'll make a start. I think there's quite a few people joined online now. I'm just going to share a couple of slides to start with. My name is Ray Owens, and I'm moderating this session today for the Instruct webinar series. So today we've got a couple of really exciting talks, one from Jeremy Keown at University of Warwick and one from Elena Seiradake, University of Oxford. Actually, both Jeremy and Elena are ex-colleagues, alumni of the structural biology division in Oxford, and their talks will have a link with the Nanobodies for Instruct facility. Just to remind everybody who perhaps isn't as closely aware of Instruct-ERIC, this is a European consortium organization which provides a single point of access to lots of exciting technology and expertise for structural biology integrated with cell and biology and biochemistry. So this includes research visits, training, internships, and some R&D funding. And the map on the left indicates the countries that are part of this consortium, really representative of a lot of the major players scientifically within Europe. Now, every couple of years, Instruct has a structural biology or integrated structural biology conference, and this year it's going to be taking place in Brussels in May, and this is just a heads up for that conference. Really encourage everybody who's interested in the business of Instruct, but also what's happening at the cutting edge of structural biology to register for that meeting. John Dolan at the Instruct Centre can provide any information if anyone is interested to follow up. And finally, the website is the best place to go to find everything you need to know about Instruct and how it works. And this is just shown here on the screen for everyone to look at. Okay, so I'll stop sharing now and pass you over to Jeremy, who's going to kick off with his presentation. So over to you, Jeremy, please.
Cheers, Ray. Oh. That's all good? Yeah, great. Cool. So thank you for giving me an opportunity to talk about some of the work that my new group at the University of Warwick that we've been doing. And today, the story I'm going to tell you about is some work that we've been doing investigating the structure and function of the CCHFV L protein. Cool. So every few years, various government and non-government agencies do these sorts of assessments where they're looking at potential causes of future pandemics. And so I've gone through these lists and I've selected out the viral pathogens here. And so when we started this work maybe 3 or 4 years ago, when I was still a postdoc with John Grimes, we were interested in understanding which potential viruses may be, or which viruses may be the potential cause of these future pandemics. And so here what I've done is I've pulled out these viral pathogens and I've highlighted the negative sense RNA viruses in bold, and then the viruses from the family that I'm going to tell you about today, which are the Bunya viruses, are also highlighted in orange. And so hopefully what you can see from these various different assessments, all of which are using slightly different criteria, is that we see many of the same viruses represented in many of these assessments. And so the three that my group work on at the moment are the influenza viruses, the Hunter viruses, as well as Crimean Congan hemorrhagic fever, which is a member of the norovirus family. And that's what I'm going to really focus on today. And so I suppose one of the major questions is, why are we interested in these particular viruses at the moment? And so excluding the influenza virus from this, really the spread of the Hunter viruses and the Crimean Congan hemorrhagic fever family of viruses is the spread of these viruses is really increasing. And so this seems to largely be driven by climate change. And so as the climate is changing, the vectors of these viruses are being spread into more populated areas. And so this is driven by the sort of changing host range of the arthropod and the rodent vectors. And so for a long time, I've been studying various negative sense RNA viruses, and I kind of like to put them into this sort of, it's kind of like a Punnett square, I suppose, to really focus our way of thinking about them. And so I like to differentiate them based on their genome, and the genome can either be segmented or non-segmented. And we also look at the site of replication and transcription, and this can either be inside the nucleus or the cytoplasm of an infected cell. And so I spent many years studying the influenza virus polymerase and how it performed these processes of replication and transcription. But today, and what my new group are mostly focused on, are this family called bunyaviruses. And so they still have this segmented genome arrangement, but rather than performing replication and transcription inside the nucleus, they perform it inside the cytoplasm. And so while the general idea of these processes is maybe broadly conserved, there are really important differences between the environment inside the nucleus and the cytoplasm. And so the family of bunyaviruses, there are around 550 to 600 individual members, and there are several really quite important human pathogens in there, as well as animal pathogens. And the one that I'm going to tell you about today is this one called Crimean Congan hemorrhagic fever virus. And so this is spread by ticks. It's quite widely distributed, found across Africa, Asia, and more recently in Southern Europe. And we're really seeing the virus turn up in places where it wasn't before. We've had infections in Spain just recently. And so one of the reasons this is such a virus of concern is that it has this really high case fatality rate, somewhere between 10 and 45%, depending on the strain and depending on the environment. Now, if we look at the general sort of life cycle of the whole bunyavirus family, they all use this sort of conserved scheme for their life cycle. So we initially have virus attachment, and this can be via a number of different types of receptors. Then have the virus that's endocytosed, and eventually this releases these genomic segments into the cytoplasm of the infected cell. Now, from here, we have the initial process of transcription to produce viral messenger RNA, and then this is translated by the host cell machinery. Now, a bit later in infection, we have this process of replication where we are able to make new copies of the viral genome, and eventually all of these new copies of viral proteins and new copies of viral genome are packaged together, and we get new viruses being formed. Now, as I've alluded to already, bunyaviruses and Crimean Congan hemorrhagic fever as well, it uses a tri-segmented genome. Now, what that means is that there are 3 different segments of the genome, and each of these encode different viral proteins. So we have the small, medium, and large subunit that are all encoding one or more important viral proteins. If we look at the architecture of each of these segments, these are called ribonuclear protein complexes. They all contain one piece of single-stranded negative sense RNA. This RNA is decorated by many copies of the viral nuclear protein, and what this does is it binds and protects the RNA from recognition and degradation by the infected cell. And then finally, sitting on both ends of the viral genome is one copy of the L protein or viral polymerase, and I'll use these terms interchangeably throughout. And so the viral polymerase is really the key enzyme in performing these 2 processes inside an infected cell. And so viral transcription, this is performed via a cap snatching mechanism. In many ways, it's likely to be similar to how it's performed by the influenza virus, where the viral polymerase is able to bind to host mRNA inside the cytoplasm of the infected cell. It then cleaves this RNA to create a short-capped primer, and it uses the short-capped primer to initiate transcription and produce viral mRNA. Now, these mRNA that are produced are very similar to host mRNA, and so they can be directly translated to create new viral proteins. Now, the other process that the polymerase performs is this one of replication. And in this process, what happens is we start off with a negative sense viral RNA. This is copied to a positive sense RNA intermediate, and this positive sense intermediate is used as a template to produce many more copies of the negative sense RNA. And so in this way, we can create new genome copies. Now, if we compare the viral polymerase from bunyaviruses to those of the very well-studied influenza viruses, we can see both some very conserved features as well as some very divergent features. Now, if we start off looking at the top here, what we can see is that the influenza virus, it has 3 discrete protein polypeptides that fold together to form the active complex. Now, on one subunit, we have an endonuclease domain. On another subunit, we have the RNA-dependent RNA polymerase domain. And then on the third subunit, we have this thing called the cat binding domain, and we'll talk about that a little bit later on. Now, if we compare the influenza virus polymerase to the hantavirus polymerase, the similarities are quite clear. We still have an endonuclease domain, we still have an RNA-dependent RNA polymerase, and we still have a cat binding domain. And these two polymerases in aggregate are about the same size. They're around 250 kilodaltons. But really the key difference between these two is that while the influenza virus polymerase is 3 discrete polypeptides that fold together, the hantavirus polymerase is 1 contiguous polypeptide. Now, many of the polymerases within the bunyavirus family are around this 250 kilodalton size, but in quite distinct contrast to many of those are the norovirus family of polymerase, and that's where Crimean Congan hemorrhagic fever comes from. So there are still similarities between the hantavirus polymerase and the influenza virus polymerase and the Crimean Congan hemorrhagic fever virus polymerase, and that you can see we still have an endonuclease domain towards the N-terminal of the polypeptide. We still have an RNA-dependent RNA polymerase, which is likely in the middle, and there is some evidence for a cat binding domain in the CCHFE polymerase. But hopefully what you can also appreciate is that it's about double the size. It's around 450 kilodaltons as a single polypeptide rather than this 250 kilodalton protein. And so really this got us started. This kind of initial question that we had is, how does the CCHFE-L protein replicate and transcribe the viral genome? And so we broke this down into 3 sort of starting research questions. We wanted to understand the endonuclease activity of the polymerase. We wanted to understand the RNA-dependent RNA polymerase activity using in vitro assays. And then finally, we wanted to determine some nice structures. Now, there was a lot of optimization, and I won't go into it today, but we eventually settled on an insect cell expression with a strep tag purification, and we could produce really quite nice amounts of protein. By mass photometry, it's largely monodisperse and quite well behaved. Now, these proteins are very big and they're often quite flexible. And so from our previous experience working with other polymerases, we wanted to look at ways very early in the process of stabilizing these proteins. And so we got some ISIDORe support to get a panel of nanobodies produced against CCHFE-L. And I'll come back to that in a moment. Now, the first piece of functional work that I want to talk about is this work looking at viral transcription. And so in this first step of transcription, what we have is the viral polymerase. It binds to host mRNA, and then it uses endonuclease activity to cleave this back to a short capped RNA primer that it can then use to produce viral messenger RNA. And so this endonuclease activity had been a little disputed or a little discussed within the literature. And so there was some work using an in vitro assay, using a virus-like particle assay, where they were able to use sequence alignments to identify the putative endonuclease boundaries. They found a conserved aspartic acid residue, and then mutation of this residue stopped the ability of the virus to propagate. And so that gave quite nice evidence that this was likely the correct region of the protein. Now, a few years later, when another group isolated and purified large amounts of this protein, they found it was catalytically inactive and did not degrade RNA. Now, this is in a bit of contrast because normally when you purify these endonuclease domains, they're very active. And so what we did is we developed a fluorescent RNA cleavage assay where we incubate viral polymerase with a fluorescently tagged piece of RNA, and we monitor degradation over time using a polyacrylamide gel. Now, we have full-length RNA here at the top, and hopefully what you can see is that over the period of about 2 hours, the RNA is almost fully degraded. Now, in contrast, if we have the RNA alone, we see almost no degradation. And so this was really nice evidence that our polymerase was active for endonuclease activity in vitro. Now, we wanted to mutate this active site because many of our downstream experiments are using short pieces of synthetic RNA, and we don't want that RNA degraded in these assays. And so we did some AlphaFold predictions with a piece of RNA as well as divalent cations, and we were able to come up with a set of residues that we believed we could mutate to block polymerase or endonuclease activity. We then inserted these mutations, produced some protein, and we were able to identify that several of these mutations blocked activity. One of them seemed to turn the activity up massively, and we don't quite understand why just yet, but we were able to identify this one residue, D693A, that blocked almost all polymerase or almost all endonuclease activity over 2 hours, which is what we need for most of our experiments. And so we now understand the sort of first part of our question, so understanding the endonuclease activity, and next we wanted to look at the RNA-dependent RNA polymerase activity. Now, I'm not going to go into too much detail here. There was a huge amount of work done by Rory, but what I just want to demonstrate here is that we can produce a nicely active polymerase that is able to perform RNA-dependent RNA activity. And so we're using a 32P radio-labeled incorporation assay. We provide the polymerase with some short RNA promoters, and then we can watch as these RNA products are produced via incorporation of radio-labeled nucleotides. Now, early experiments, we had quite weak activity. We were producing large amounts of the shorter product, and then through lots of development, we were able to develop this assay so that we can produce now really nice amounts of full-length product. And so that was great. And the reason I suppose we want to be doing this is our ultimate goal is to try and understand how we might inhibit the activity of the polymerase or inhibit the activity of the endonuclease, and so that we can stop viral infection and maybe develop some new antivirals. Now, with this sort of goal in mind, we began some quite low throughput screening using our 32P assay. And really the only major hit that we came across for stopping RNA extension was this compound called acyclovir. And it quite potently inhibited extension at the highest concentrations. This was very unexpected. This is a well-documented drug that's exclusively used against DNA virus polymerase. And so we're not really sure why this compound works. We don't believe that there are other examples in the literature of RNA viral polymerases being inhibited by acyclovir. I'd be very happy if someone showed me that I was wrong. That would be great. So this is quite an odd functionality that, again, we don't really understand. Now, our 32P assays are fantastic, but they're quite limited in their ability to scale. And so really where we're moving with this assay now is to develop single molecule fluorescence by labeling both a radio or a fluorescently labeled primer, as well as some of these template RNA, and we can monitor extension. And so this is where we're heading with a lot of this work now. And so now we've done, we hopefully understand a bit of the biochemistry of our polymerase, and we understand how these various activities work. Now we wanted to really use this information to move into determining some structures. Now, the first structure that we determined, this was done at the OPIC facility up at Struby, and we were able to determine an RNA-free bound polymerase structure to about 2.8 angstrom. I'm just going to skip almost entirely over the structure because there's a more interesting one that I would like to show you. Now, we know that the polymerase's role is really binding the termini of the genome. So both the 5' and the 3' end are likely associated with the polymerase. And so we developed this electromobility shift assay to try and screen for conditions where we can get nice amounts of binding of RNA to the viral polymerase. Now, hopefully what you can appreciate is that only under quite high RNA concentrations and quite low salt concentrations will we be able to get nice binding to our polymerase. Now, here is the structure that we were able to produce. This was collected at EMBL Heidelberg, again supported by ISIDORe. And so here we have the 2.1 angstrom structure of the Crimean Congan hemorrhagic fever virus L protein. And I'll just talk you through the interesting bits of this structure now. So we have approximately 2,000 amino acids in our structure. We're missing quite a large region in the middle. And starting from the N-terminus, what we observe is the endonuclease linker region, the vRNA binding lobe, as well as the fingers, palm, thumb ring, and lid domains. And these are all enlarged compared to their homologs in other species. We observed two new insertions, one of these insertion 1 and the other insertion 2, and we only observed part of one of these. Now, if we look in the active site, we see all of the canonical residues that we expect, motifs A through F, important for nucleotide and RNA binding. Now, really quite important for this structure is the presence of this nanobody 20,096 that we observed and helped us solve preferential orientation. We have a couple of binding sites. This is the first zinc finger. These are non-catalytic metal ions, and we think that they're involved in rigidifying the polymerase core. So we have 2 zinc ions as well as 1 magnesium ion. There's also an additional magnesium ion in the center of the active site. Now, the most interesting part of the structure is that we observe this very ordered piece of RNA packed into this pocket. And so what these are the 13 residues from the extreme 5' termini of the genome. And really what this does is this serves to tether the genome into the polymerase so that while the polymerase is reading along the rest of the RNA, it always maintains it, or it always is associated with the viral polymerase. Now, because we have an RNA bound and an RNA free structure, we can look at the differences that the RNA binding causes. Now, the first of these is that we see the ordering of some quite large regions of the viral polymerase, and these fold and pack over this piece of RNA, really holding it tightly within the polymerase core. We also see stabilization of residues inside the active site polymerase. And so what we've been able to demonstrate, and we've done some functional work to show this as well, is that binding of this RNA really turns up the activity of the polymerases and is probably absolutely required for good activity. Now, because this is a bit of an Instruct talk and we had some nice nanobodies produced for us, I thought I'd briefly go on a bit of a divergence about them. So we were able to end up with 35 nanobodies. These were from 19 different families, or from 31 different families. And really we had this problem of which ones should we be progressing with to try and solve some of our structural biology problems. Because really experimentally determining that many structures is not something that we were that keen to do. And so after a trip to Bristol to visit some colleagues, there were some people talking about AlphaFold predictions of nanobodies binding to various or their targets. And so I set a very talented undergraduate student to see if they could discern some nanobody binding sites. We looked at both full-length structures as well as targeted domain truncations. And we were able to get AlphaFold to confidently predict 13 of our 35 nanobodies bound to the polymerase. Now, 12 of the 13 were binding to exactly the same site. And so that was nice. And where we have determined some nanobody polymerase complexes of both some of these 12 confidently predicted nanobodies, there's essentially perfect agreement with the AlphaFold prediction. So while this doesn't tell us where all of the nanobodies are binding, it certainly truncates our list down from 35. And so we don't need to now investigate all of these 12. So while the affinities may be a little different from a structural biology perspective, they all seem to be quite similar. And so just to go back to our structure for a moment, the polymerase core that we've solved, some 2,000 residues. This is bigger than most full-length bunyavirus polymerase, but we're actually still missing 2 very large regions at both the N and C terminus that are containing, there's at least 3 functional activities in there that we haven't quite got to yet. And so hopefully what I've convinced you is that we now understand the endonuclease activity of the enzyme. We understand a little bit more about the polymerase activity and how we may use this for antiviral design and development. And we've begun to produce some CCHFE-L protein structures. And really where we're heading next is we want to determine some more specific functional conformations of the polymerase to understand the sort of details of all of these steps. So this has really been the first project that my group at the University of Warwick have done, and thanks to my team, collaborators Nicole and Rory here at Warwick, as well as John, Loic and Franzi in Oxford, and Jan and Els at the VIB for nanobody production. Thank you.
That's great. Thanks very much, Jeremy.
That's right.
If you stop sharing, I can open the floor to questions, please. If you've got questions, drop them in the Q&A. Nothing appearing yet, but let people have a few minutes to think. While that's happening, an obvious question, Jeremy, with your nanobody, presumably you've looked to whether there's any functional effects of adding them into the system.
Yeah, so we are currently trying to work through this. CCHFV is a Cat4 pathogen. So we are in the process of getting the virus-like particle system here to Warwick to then screen how they may be functioning. We are similarly putting these through our various in vitro assays at the moment. And we are talking, once we've done some of these more in vitro or VLP style assays, we're going to hopefully put them into the Cat4 virus systems and see what they do. I think either for using the nanobodies themselves as some sort of therapeutic or identifying sites on the polymerase that we should be targeting with maybe more traditional antiviral approaches. Cheers.
Great, thank you. There's one, someone's got their hand up, but could they put the question in the chat? I think John's just asked that, please. Just so we can handle it through the chat rather than opening it up otherwise. Let's wait on that one. Well, if I guess, John, Jeremy, you'll pick questions up afterwards if people can email you. Is that okay if they don't actually talk about this? Oh, Elena's dropped a question out. Oh, here's a question. So sorry, I've got the questions now. First question from the person who had the hand up first was, are you able to detect multiple conformations of the polymerase?
So we see some low resolution of the missing bits. We do see low resolution envelopes at lower thresholds in the map, but nothing that we can model at the moment. We're hoping that by adding in some of these RNA or some of these nanobodies, we can trap the polymerase in specific conformations and order the whole thing. We've not been able to do that yet.
Okay, thank you. Eleanor, you had a question. Could you put it in the chat or just put it up? Bit of a time lag here. Eleanor, did you want to just speak your question? Because I know you've obviously got the mic.
Talking to me, Ray?
Yeah.
Oh, sorry, I pasted a question in the chat. I wasn't sure if you can read. It's just a really technical one because we're also working with nanobodies, finding them really useful. And I know it's overwhelming. You get so many. What technique do you use to even know which one is a high binder? Obviously, that's really hard to get out of AlphaFold.
Yeah. So we haven't gone too much into affinities. I know that there are vast differences in the affinities of various nanobodies. We have used the sort of idea that in order for the nanobody to make it through the discovery phase, they're probably either very low micromolar or somewhere in the nanobody range, or sorry, nanomolar range. And so we haven't done one of these studies. I think there's about to be a feeder bio, which is about to come to the department. And so I think we'll play with that because we don't have to do too much additional labeling, as I understand it. SPR is doable. The polymerase are quite unstable, so we can't load the chip up with them permanently. There are some technical hassles there that we're trying to get away with label-free approaches. Yeah.
I mean, I can share my best experience, but I'd really like to stay in touch. So if you get something even easier, that would be better. We've been using ELISAs a lot. Like we found they work well in high throughput, and as long as you've got a his tag or something when you're nanobody.
We have an ELISA that we're setting up for a different system.
Yeah, but there might be even better ways. I don't know. That's our current best.
Well, just to chip in, Eleanor, we also use ELISAs a lot for the first pass screening. As you say, it's convenient and easy to do, but it's not, then we'd go on to BLI or something like that for soluble proteins.
Yeah.
There's another question popped up from Marques de Silva. He said, good presentation. I'd like to know if there's a huge difference between AlphaFold and your cryo-EM map.
Not massively. I think there are some flexible regions that we know that in response to RNA binding, they rearrange. And so these are changes within the active site of the polymerase. In broad strokes, no, it's quite similar. It's very confident in the region that we can see, and it confidently predicts the flexible regions, but it's not confident in their location. So it knows domain boundaries, I suppose, is the comment there.
Just remains for me to thank Jeremy and Eleanor for some fantastic talks, and also thank you for everyone who joined today, and I hope you had an interesting time. And John, is there any final words from you?
No, that's all great. Thank you, Ray, and we'll see you all at the next webinar.
Okay, bye now.
Thanks, everyone.